just kind of go through step by step of mitosis and meiosis. So some main takeaway from here. You don't need to necessarily know prophase, metaphase, anaphase, and telophase in the form of PMAT. It's good to know it, and when I use those terms, you know what I'm referring to. For you guys, just know what happens, what needs to happen before the next thing happens. So what I mean by that is, for example, okay, before there can be any type of nuclear division where the nucleus is dividing, right, into both of the cells, that DNA has to replicate first, right? If you don't replicate, then you're not going to have the exact same number of chromosomes or genetic material in both of those cells, okay? And you'll also notice that in mitosis, you start with a diploid number or diploid nucleus, and the two new daughter cells are genetically identical. So you also end with diploid cells, okay? All right. Also in mitosis, right? The other thing you need to take a look at here is you, at some point, right, when the chromosomes line up beside each other, which is under the phase called metaphase, all right, you can see that these long string things called spindle fibers, they connect to the chromosomes, all right, and they're going to split them apart. Now we give them these special names. When they've replicated, we call one half a chromatid or sister chromatid, and the other one a sister chromatid. So you have sister chromatids here, and it's the sister chromatids that get split from each other. Okay, in doing so, you result in two daughter cells with the exact same number of chromosomes that you started with. So you start with a diploid cell, you end with a diploid cell. Okay, now I want you to evaluate the use of mitosis to make a new offspring. So basically making gamete sex cells. Would mitosis be a good idea to create gametes? Well, let's take a look. So let's say then your father in his sperm gives you 46 chromosomes instead of 23, and your mother does the same thing, 46 instead of 23. The zygote will produce would have 92 chromosomes. That is insane. And then if you don't ever go in half that number of chromosomes, like if you never make a haploid or gametes, cells that are half the number of the original, then the 92 chromosomes found in that sperm will then go and combine with or fuse with the 92 chromosomes found in the nucleus of the egg. And the next up, you'd have an individual with 184 chromosomes. That would be impossible. You're like no longer human. I don't even know that would be even doable, right? So this would be horrible. So we don't want to use mitosis to make new offspring. So we want to use something called meiosis. All right. In this case, you've got a haploid number in the egg and a haploid number in the nucleus. All right. So half the number of the chromosomes. You fuse them together, and then you have a diploid number, and that is the zygote. Okay. Now this is good. This means that every time you have a new zygote, it's always the same number. Always for humans ends up being 46. Okay. In meiosis, it looks slightly different from mitosis. You can see here that at the beginning, everything looks the same. You always have to have DNA replication happen before nuclear division can happen, except that a little something else happens, right? In meiosis, the chromosome from your mom and the one from your father, the one that's more similar in shape and size, they will pair up beside each other. And that's what you call homologous chromosomes, okay? So in humans, you'd see 46 chromosomes lining up beside each other, making two pairs across that are similar in size and structure. Because this one only has two chromosomes, it's nice and easy. We can just pair those up beside each other. We assume that those are the homologous chromosomes. And then the next thing will happen is not the sister chromatids this time, but the whole homologous, the whole chromosome in the pair will split. But before that even happens, you're going to notice that the genetic material is slightly different, okay? So originally you saw with the whole red one, now you have red with a little bit of blue. So what's happening there is something called crossing over. That's just like in the next image, okay? So in here you can see if this was one chromosome and this is the other one, they end up, actually let's do it like this. They end up like coming together, and they're going to exchange some genetic material. So eventually when they walk away and are split up, you end up getting a mixing of genetic material from the mom and the dad's chromosomes. This is important. This is why our sex cells are going to be slightly genetically different from each other, making all this providing all this variation in the offspring that it's produced. Okay, so next little bit that's happening here. What we want to make sure happens next is Before, it was the homologous chromosomes that split apart. Now it's going to be the sister chromatids. So they're going to split up, and this ensures that the number of chromosomes in the gametes in the sex cell is half the number from the original parent cell. Okay, this is good. This means that now in the gametes, whether these are 4 sperms or 4 eggs, you have lots of variation, okay? And then there's a new combination from the mom and the dad's chromosomes. Okay, so that's all I want you to know for that. And the next little bit's going to be on mitotic index.